Hi students, have a good time. You are welcome to Biology O Levels IGCSE 610. We are going to discuss chapter 5. And chapter 5 of Biology O Levels IGCSE is about enzymes. Perhaps you might have studied in your pre-O levels and in early classes that there are certain catalysts inside the cell and in some living systems which are called enzymes. Let's define enzyme. What is an enzyme? Enzyme is simply called biological catalysts. If I define the two words biological and the catalysts, the simple meaning is that they are produced only by the living things. It does not mean that they work only inside the living things, but it means that they are produced by the living things. They can be working inside the living cells, and we can extract them from the living cells or the living organisms, and we can make them work in laboratory conditions. So the biological means that they are produced by the living things, means they cannot be synthesized artificially in some factory or in some industry. We cannot produce them synthetically. They are produced only by the living things. And the second word is catalysts. What are catalysts? You might have studied in chemistry or you might be familiar with them, that a catalyst is a chemical which speeds up or which alters the speed of some chemical reaction. This is true for inorganic chemicals, inorganic catalysts, but for biological catalysts, the thing is that they are speeding up chemical reactions. What does it mean? Why is there a need of speeding up of chemical reactions? Basically, in living cells, in living things, most of the chemical reactions, they are either very slow or they are multi-step. And it's very difficult to proceed them without any catalyst. Say for example, I'm going to write here one example, that if there is a chemical called starch and we want to react it with water. This is very simple reaction which is occurring in our mouth or in small intestine, digestion of the starch. This starch will be digested when water is added. These two are the reactants, starch and the water. And the product is maltose. But if we are doing this reaction at body temperature of about 37 degrees Celsius, a few gram of the starch will take several hours to be digested into maltose. And with such a slow reaction, life cannot sustain. We cannot wait for such a long time to let starch digest so slowly, because this will support a very slow metabolic rate. But the living organisms, they need a high metabolic rate. So the other choice is either to increase the temperature up to 100 degrees Celsius. We can speed up this reaction. Means it's very simple that if we will increase the temperature from 37 to 100 degrees Celsius, these reactants will be colliding with each other very quickly and the rate of reaction will increase. But once again, our body cannot sustain in such a high temperature. So what's the next choice? Either we will have to wait or we will have to increase the temperature. The third choice is use of enzyme. There is an enzyme called amylase. If this amylase enzyme is present, starch will react with water in the presence of enzyme amylase and maltose will be produced within few minutes. Say for example, when you are chewing some food of the carbohydrate such as wheat and rice or barley or oat, while you are chewing your bite, a lot of starch is digested, and the rest of the starch is digested in your small intestine, means the reaction will become very fast. So there is a need of catalysts always in the living systems to speed up the chemical reactions. Although non-biological catalysts, the inorganic catalysts, they are sometimes speeding up the reactions, sometimes they are slowing down the reactions. But here in biological system, we need to speed up the chemical reactions always. So I think it's very clear to you what is meant by the biological catalyst. This is the definition of enzymes, and this is also one of the basic features of enzymes, that they are the biological catalysts. Okay, let's see the next. The next feature is that they are protein in nature. About 99% of the enzymes are protein in nature, although there are some enzymes which are nucleic acids in nature, but about 99% of the enzymes are protein in nature. You have already studied the biological molecules in O levels, that the major chemicals, the macrochemicals in the living systems are the carbohydrates, proteins and lipids. So the proteins are making the enzymes, or enzymes are protein in nature. Okay, what type of proteins they are? They are globular proteins and they are water soluble proteins. Two basic features of the proteins are that they are almost spherical or round in shape and they are soluble in water. These type of proteins are the enzymes. Means they are not structural proteins, they are not fibrous proteins, and they are also not insoluble in water, rather they are soluble in water. It means all the chemical reactions which are catalyzed by the enzymes are possible only when the reactants are dissolved in water. So are the enzymes, that they are soluble in water. Okay, so this is the second feature of the enzymes, that they are protein in nature. The third is that they are always larger than the substrate. So what is this word substrate? Any chemical which is acted upon by the enzymes is called substrate. Here in this case, which we have studied, starch plus water, they are reacted with each other to be changed into the maltose by the use of enzyme amylase. Here in this example, starch and water are the substrates and amylase is the enzyme. So there are the two things. There is one thing is enzyme and the other thing is substrate. Means this is enzyme plus this is substrate, and they are changed into enzyme plus product. So what I am discussing here, that they are always larger than the substrates. In your advanced levels, in your higher studies, you will study that the basic job of enzyme is just the guidance of the reaction. They are not themselves involved in chemical reaction. Means if there is starch and there is water, it is automatically reacting. Starch is automatically reacting with water and it's automatically being converted into the product, that is maltose. So enzyme is neither the reactant nor the product, but it's just the facilitator. Just for example, if there is a little baby who wants to clap and is unable to clap, someone elder can guide the baby how to clap, how to collide in proper direction. So the job of enzymes is to let the substrates collide with each other in a proper direction so they can undergo the reaction. So its job of the enzyme is the guide, and it's very logical that the guide must be bigger, must be larger. Such as if there is a little baby of 3 years Another baby of 2 year cannot guide that baby of 3 year. Someone elder who is 10 year or 15 years old, elder than that 3 year baby, can guide that baby. So is here that this is the baby and this is the guide. Enzyme is the guide and substrate or the substrates are like the baby. So it is clear that substrates are always smaller than enzymes and enzymes are always larger than the substrates. Okay, another feature is that enzymes have specific 3D shape, three-directional shape. If we look at the molecular structure of some enzyme, they are not like linear chains. They are not like fibers. Rather, they are of specific 3D shape. And this specific 3D shape is very important for their normal action, for their normal activity, for the normal catalysis activity. If normal 3D shape of the enzyme is not there, if there is any change to a specific 3D shape of the enzyme will not be able to do the catalysis, the specific catalysis. So all the enzymes, they have their specific 3D shape. I am not saying that all the enzymes have the same shape. Each enzyme has a different three-directional shape, but whatever the shape of one specific enzyme is, it is specific. Amylase is of specific 3D shape and pepsin in the stomach is of another 3D specific shape, and the trypsin in duodenum is of another 3D specific shape. And that 3D specific shape is very important for the normal working of an enzyme. Okay, then another feature is enzymes remain unused. Means, once again, as I have told, the basic reaction is not the way that enzyme will be participating in it, in the sense that either it is formed or it is consumed. As I have told here, that enzyme is just the guide. When the guidance is over, the job of enzyme is over. Substrate and one substrate or more than one substrates, they will be reacting with each other and enzyme is just guiding them. And when the substrates are converted into the products, there is no more need of enzyme and enzyme will remain unused. I am not saying that enzyme will remain unchanged, although when it will be participating with it, the enzyme is holding the substrate or more than one substrate, it will undergo certain changes and then enzyme will reverse its changes, but that reversal of the changes in the structure of enzyme might not be 100%. Means there can be a minor change in the structure of enzyme, but overall enzyme will remain unused. It might be changed to some extent, but it will remain unused. What does it mean? That enzymes can be used again and again and again. Say for example, if there is a little saliva secreted in your mouth and you are chewing a loaf, a bite for 30 seconds or for 40 seconds, this enzyme will go on working repeatedly. The starch molecules will be digested again and again, different starch molecule. One enzyme will occupy the substrate, product is formed. This enzyme will come back to capture another substrate to catalyze it. But it does not mean that once enzyme produced in some part of the body, they are sufficient forever. I am not saying that once saliva is secreted in your mouth will be sufficient forever. Whenever you will eat, the saliva secreted already in your mouth will be there. No, every time we are producing a new saliva, we are refreshing, because the older enzymes, after a few catalyses, they are exhausted. They become less active. They slow down in their catalysis. That's why we are producing new enzyme, but it does not mean once again that enzyme is used just once. They are used repeatedly, again and again and again they can be used. So I have said that enzymes remain unused in the chemical reaction. Another thing I have written here as a basic feature of the enzymes, that enzymes are very fast acting. They are not slow acting. What does it mean? That we have to speed up the chemical reaction. If enzymes are working slowly, then we cannot speed up the chemical reaction. They are basically the facilitators. They have to facilitate the process. When some process is facilitated, the process is done quickly. Say for example, if you are solving some sum of the mathematics or some other subject and it's taking a lot of time to you, but if some teacher of yours will facilitate you, will guide you, then you will do the question very quickly. So the job of facilitation is to speed up the process. So the facilitators are fast acting. They are acting very quickly. One of the world's fastest enzyme is catalase. Catalase enzyme can break hydrogen peroxide molecules into water plus oxygen with such a speed that several million molecules of hydrogen peroxide, they are broken down into water and oxygen in a short while, within seconds it can be, because this is the byproduct of respiration, cellular respiration. Everywhere it's being produced in all the living organisms. So every living organism is producing this enzyme catalase, and as this hydrogen peroxide is toxic chemical, it's a strong oxidizing agent, so it must be broken down into harmless chemicals, water and oxygen, and that's why this hydrogen peroxide is very quickly converted into water and oxygen by the enzyme catalase. So enzymes are very fast acting. Another feature is that they are easily affected by the temperature and pH. I have said that they are unused, they are fast acting, they are of specific 3D shape, but they are also affected by some factors like temperature, like pH, and many other factors such as poisons, inhibitors, and even there are some helping chemicals which can help maintain the structure of enzyme. So one of the two basic factors which are very easily affecting the enzyme structure and their speed of catalysis are the temperature and pH. So these are just a few of the basic features of enzymes, that they are the biological catalysts, they are protein in nature of 3D shape, they are larger than the substrate, they are of specific 3D shape, and they remain unused. They are very fast acting and they are easily affected by temperature and pH. Apart from these basic features, there are some other points related with the enzymes, that is, that there are some enzymes which are breaking the substrates, larger substrates into smaller substrates. This type of chemical reaction is called catabolism. The breakdown of larger molecules into smaller molecules is called catabolism. So there are some enzymes which are catabolic in nature, while in some other chemical reactions, small molecules are joined with each other to make the larger molecules. This type of reactions are called anabolism, and there are some enzymes which are anabolic in nature. Breakdown of the starch is a catabolism, and joining of glucose molecules to make glycogen in our liver, or joining of glucose molecules to make starch inside the plants, is anabolism. Joining of amino acids to make proteins is anabolism. So enzymes are catabolic in nature, they are anabolic in nature. Moreover, there are certain enzymes which are working only inside cells. They are called intracellular enzymes, such as Respiratory enzymes, they are working inside the living cells. They are speeding up chemical reactions, but those chemical reactions which are occurring inside the cells. That is cellular respiration or oxidation of food. So these chemicals are called, these enzymes are called intracellular enzymes. While other enzymes are those which are working outside the cells. Means cell will secrete the enzymes out of them, and they will be working in between the cells in some cavity, such as enzymes working in your mouth cavity. They are extracellular enzymes. They can also be called intercellular enzymes. So there are intracellular enzymes and there are intercellular enzymes. I have discussed two words, anabolic enzymes and catabolic enzymes. Two words, and I have discussed two other words, that they are intracellular enzymes and there are intercellular enzymes. Those which are working in between the cells, or they can also be called extracellular enzymes. Extracellular enzymes or intercellular enzymes and intracellular enzymes. The enzymes working inside the cells. So these are some of the basic concepts related with the enzymes for your O-levels biology. Then next we have to study in this chapter how is temperature and pH affecting the rate of enzyme controlled reaction. Thank you.